minutes graduation.
Good morning, everybody, and welcome to this session. I open the academic ceremony in which Justine Webster will defend the academic thesis, The Role of Pre-Receptor Glucocorticoid Metabolism in Chronic Inflammatory Disease-Associated Muscle Atrophy. I practiced that. May I invite you to first present a summary of your research and the conclusions and recommendations, and you'll have about 15 minutes for that.
Okay, thank you. Just to reiterate, my name is Justine. I'm here to talk about my thesis, The Role of Pre-Receptor Glucocorticoid Metabolism in Chronic Inflammatory Disease-Associated Muscle Atrophy. So this thesis focuses on muscle wasting in the context of chronic inflammatory disease. So chronic inflammatory disease is an umbrella term for several diseases, such as inflammatory bowel disease, chronic kidney disease, chronic obstructive pulmonary disease, and rheumatoid arthritis. And these diseases all have a common feature of low-grade inflammation with flare-ups of disease. And this thesis primarily focuses on COPD and rheumatoid arthritis. And World Health Organization attributes to up to 60% of causes of death worldwide to chronic inflammatory diseases. So COPD is a progressive lung disorder characterized by chronic inflammation and irreversible remodeling of the airways, and that leads to airflow limitation. There are two pathological processes that underpin this remodeling, including chronic bronchitis, which leads to the narrowing of the airways, and emphysema, which leads to the alveolar enlargement. Many COPD patients have a combination of both chronic bronchitis and emphysema features. There are several risk factors associated with the development of COPD, such as smoking, chemical exposure, and pollution. In addition, COPD is also associated with increased risk of hospital admissions, reduced exercise capacity, and contribute to a third of cause of death worldwide. Acute exacerbations of COPD are defined as episodes of intensified disease symptoms, usually as a result of bacterial and viral infections, but not always, and often accompanied by systemic inflammation or pulmonary inflammation. Acute exacerbations often lead to the acceleration of the disease progression and correlate with poor prognostic outcome for these patients. Another chronic inflammatory disease that this thesis focuses on is rheumatoid arthritis, or RA. It's a chronic systemic autoimmune disorder characterized by progressive joint destruction, loss of cartilage, synovitis, joint inflammation, and bone deformity. So the immune cell infiltrates into the synovitis of the joint, then leads to a pro-inflammatory cytokine and chemokine secretion, which then paves the way for more inflammation. Affecting 1 to 2% of the population globally and disproportionately affecting elderly patients and women, joint destruction in RA results in bone deformity, joint swelling and pain, and is incredibly debilitating for these patients. It leads to poor mobility, increased risks of falls and fractures, and increased risks of hospital admissions, which is ultimately a poorer quality of life for these patients as well. It's also associated with the disease flare-ups. So both COPD and RA are chronic inflammatory diseases and have distinct pathophysiologies. However, like many chronic inflammatory diseases, they are associated with disease flare-ups and exacerbations. They are often accompanied with systemic and local inflammation. This is treated with glucocorticoids, therapeutic glucocorticoids. and have systemic and peripheral complications of the disease such as muscle wasting. And we know that inflammation and therapeutic glucocorticoids are known drivers of muscle wasting as well. So skeletal muscle is one of the largest tissues in the body. It comprises about 40% of the total body mass, and its main functions are to provide support and locomotion to the skeleton in the body, but also play a crucial role in energy metabolism and glucose homeostasis. So skeletal muscle wasting is observed in about 20 to 40% of COPD patients and up to 40% in RA patients, and leads to a reduced physical activity, and again is associated with increased hospitalization, increased risk of flare-ups, and increased mortality. In healthy tissue during homeostasis, muscle tissue is continuously renewed by a fine balance between muscle building and muscle breakdown. Several processes such as muscle protein turnover, including muscle protein synthesis and degradation, and myonuclear turnover, including myonuclear degradation and accretion, are involved in maintaining this balance. But stresses such as, and stimuli such as exercise, disuse, repair, disease, aging, change this balance. So in response to stimuli such as exercise, the balance would be tipped in favour of building, and we see an increase building of muscle fibre. In response to disease, aging, disuse, the balance is tipped in favour of wasting, and you see this reduction in muscle fibre size. So inflammation has been implicated as a potential direct cause of muscle wasting, where inflammatory cytokines directly activate the muscle wasting pathways. Therapeutic glucocorticoids are often used to treat the inflammation. However, therapeutic glucocorticoids have also been implicated in driving muscle wasting in themselves. Besides therapeutic glucocorticoids, glucocorticoids are also produced endogenously. Inflammatory cytokines are known to activate the hypothalamus in the brain, which leads to the adrenal cortex releasing glucocorticoids, or cortisol in human, corticosterone in mice. And these elicit their anti-inflammatory effects to resolve inflammation in the body. Local activation of glucocorticoids in the body is modulated by the 11-beta hydroxysteroid dehydrogenase 1 enzymes. When inactive glucocorticoids reach skeletal muscle, where this enzyme is found in abundance, they are activated to their active counterparts, so cortisone to cortisol in human, or dehydrocorticosterone to corticosterone in mice. We know that inflammation increases the activity and the abundance of 11-beta HSD1 in the muscle, which leads to increased glucocorticoids in the muscle. So we hypothesize that if we deleted 11-beta HSD1 in a model of COPD, we would be able to reduce the amount of active glucocorticoids being activated in the muscle, and this may alleviate the muscle wasting we see. To do this, we utilized a model of acute exacerbation of COPD. We had 11-beta HSD1 global knockout animals that were intratracheally instilled with emphysema to induce, elastase to induce emphysema. And we also then intratracheally instilled them with LPS, which is a bacterial component, to induce a pulmonary inflammation. And we saw here that with the emphysema development in the wild-type animals with elastase and the infiltration of immune cells into the lungs with an acute exacerbation. And these were similarly seen in the both wild-type and 11-beta HSD1 knockout animals. We also looked at micro-CT hind limb analysis of the muscles and the muscle weights of these animals, and saw that in an acute exacerbation of COPD, we had significant muscle loss in both wild-type and 11-beta knockout animals, but it seemed to be marked more in the knockout animals. We then used in vitro analysis, and looked at the application of corticosterone, which is a glucocorticoid, and dexamethasone, which is a therapeutic glucocorticoid, and the effects that this has on cells in vitro. And we saw that muscle building was impaired when we added in these steroids. We then wanted to know what the glucocorticoid administration is known to induce muscle wasting in rheumatoid arthritis. We wanted to know the role of the glucocorticoids. So what we did is had a TNF transgenic mouse model of polyarthritis, or RA. We treated these with either vehicle or corticosteroids or glucocorticoids, and we saw that in the bone of the rheumatoid arthritis mice, as shown in yellow, there was infiltration of inflammation that was abrogated when treated with glucocorticoids. However, the muscle of these mice, we saw that the cross-sectional area, which is the fibre size, was significantly reduced in the glucocorticoid-treated polyarthritic mice. And this was also shown when we looked at the muscle weights. With glucocorticoids and polyarthritis, there was a significant decrease in the muscle. So then we wanted to look at the effects that 11-beta-HSD1 has in mediating this glucocorticoid-driven muscle atrophy. So if we deleted 11-beta-HSD1, could we reduce the muscle atrophy? So again, we utilised the rheumatoid arthritis mice with 11-beta-HSD1 deletion and treated them with glucocorticoids. And similar to the previous slide, we see this reduction in muscle weight in rheumatoid arthritic mice with glucocorticoids. However, the mice that had rheumatoid arthritis without the 11-beta-HSD1 enzyme, we saw there was a protection of muscle wasting in these mice when treated with glucocorticoids therapeutically. We also used ex vivo analysis of muscle biopsies from the wild type and the 11-beta-HSD1 mice, and we added TNF, which is a pro-inflammatory cytokine, to see what the wasting gene expression would be, and saw that it was indeed induced with glucocorticoids. However, when we added DHC, which is dihydrocorticosterone, remember the inactive glucocorticoid, which is no longer being able to be activated by the 11-beta-HSD1, there was no significant induction in the wasting gene expression as we saw in the wild-type mice. But then when we combined the inflammation and the glucocorticoids, this induction was restored again. So to conclude, when we had 11-beta-HSD1 inhibition in a model of acute exacerbation of COPD, muscle wasting was aggravated. When we had glucocorticoids treating rheumatoid arthritis, muscle wasting was aggravated. But when we combined glucocorticoid therapy with the 11-beta-HSD1 inhibition, there was a partial protection against this muscle wasting. So it may hold promise that adjuvant therapies of glucocorticoid therapeutically and 11-beta-HSD1 inhibitors may be able to prevent this induced muscle wasting in chronic inflammatory disease. Thank you.
Thank you for that very clear summary and well in time. The opposition will start with Professor Welting. He's a professor of molecular cartilage biology at this university, and he was the chair of the manuscript assessment committee.
Thank you, Pro-Rector. So, dear candidate, congratulations with the finishing of your thesis, and especially I think because you're aiming for a double degree, so let's see whether that will be finalized today.
Yes.
And of course also my congratulations to your team, which is here live and digitally. So I would like to take you actually to chapter 5. So chapter 5 I think is a really interesting study where you are looking at the polyarthritis model, the transgenic TNF model. Mm-hmm. And what I particularly like about actually that chapter is that it's like multidisciplinary in terms of the types of tissues and the types of cells that you have been investigating. So I think that is a really nice broad overview to look at a disease that has a systemic, very highly systemic character. So before diving into the deep, I would first like to actually ask you, where are the data in chapter 5 that are supposed to be in figure 1 and 2?
Let's have a look at figure 1 and 2.
This is a bit of a nitty-gritty question because your figure actually starts with number 3.
Ah, I see.
So this is evidence that I actually read your chapter.
Yeah, yeah. So figure 1 and 2 is primarily the bone data and the data that I can't believe I missed that. The bone data and the data that corresponds with the disease progression in these mice. So what we did is we looked at the TNF transgenic mice and the disease progression in them, and if the glucocorticoids actually reduce that disease progression. So those were primarily seen in figure 1 and 2.
Well, thank you. So actually, throughout that whole chapter, you're talking about anabolism, catabolism, metabolism. You're actually also measuring a couple of genes. So one of them is called RAD1, and 3 others are called FOXO1, TRIM63, and FBXO32. Actually, you categorize them as anti-anabolic and catabolic. What's the difference between anti-anabolic and catabolic? Because to me, it actually sounds the same.
Yeah, of course. Thank you for your question. So the difference between the anti-anabolic and the catabolic processes is the catabolic processes are primarily driving the wasting, so the breakdown of the skeletal muscle, whereas anti-anabolic are sort of the gatekeepers of not driving any anabolism. So whilst you have like the fine balances that were on my slides, with catabolism, you're going to be reducing the muscle waste, like reducing the muscle, whereas the anti-anabolic is just not going to be recovering it. So if you have anti-anabolic and catabolic, you're going to have muscle wasting because you're not having this anabolic driver to balance that off.
Okay, yeah. Thank you. Then going a bit deeper, so this mouse model that you use, so what I've understood from it, it is a whole body transgenic TNF mouse model. So these mice are being born, and at some point in time, they start to develop deep body arthritis, right?
Yep.
And then you write, actually, in your thesis that you started administering the glucocorticoids in the drinking water at first measurable onset of arthritis. So how did you actually determine that? So what are the signs that you're actually using in order to reproducibly actually determine the moment that you need to start administrating the glucocorticoids?
Yeah, of course, thank you. So the onset of the clinical scores that you would have seen in figure 1, should it be there, that we looked at was things like measurable joint deformity, food intake, body weights, the kind of clinical measures that you would see typically in patients with rheumatoid arthritis. So
what stage in rheumatoid arthritis, in human rheumatoid arthritis, would that be?
So this would be a very early onset.
Right.
So it would be comparable to human cohorts. It would be very first signs of sort of like joint inflammation. Obviously, we also looked at things in the serum like CRP, IL-6. Those are obviously indicators in patients with rheumatoid arthritis.
You can also find that in these mice.
In these mice as well, yeah, exactly.
Right.
Yeah.
Okay, great. Okay, so then actually we go to page 156 and further, because there's where the fun starts for me actually. And so there is a protection, if I'm not mistaken, of the loss of trabecular bone, right? When you treat these TNF mice with the corticosterone. While then in the, so it has a protective effect, and actually you did by micro-CT and you determined that this is not the thickness of the trabeculae that is somehow affected, but the number, right?
Yep.
If I then go to the data in figure 5, so these are a combination of in vivo and in vitro data, if I'm not mistaken, then you measured measures of bone formation and also measures of bone resorption. And actually both of them are like negatively affected by the glucocorticoids. So for example, osteoclast pit formation is reduced, but also, for example, here, I think it was ALP expression or P1 and P, so this is the pro-collagen, right, is also being reduced. So I'm a bit actually confused now, because on one hand you show a protection of bone loss, but on the other hand you actually show that both processes that are involved in bone homeostasis, so the making and the breakdown, are both negatively affected. So how do you think that works in vivo?
Thank you for the question. I will be completely transparent here and say with the bone work, a lot of the bone work that was done was from one of my esteemed colleagues, Chloe Fenton. So what we did is essentially I did the muscle side of the analyses, and she did the majority of the bone analyses. With that in mind, we still know that in this particular study, there was this, as you said, on figure, was it 5 you said?
It's chapter 5 and figure 5.
Figure 5.
Yeah.
When you see these inductions in these, like ALP and PI, but we see the inductions but not as much in the treated mice. I'm not really too sure why that is. I think it's the balance between the osteoclast reformation and osteoblast reformation, but yeah.
And because I think one of the conclusions was somewhere at the end of the chapter that you think that it is osteoclast actually activity that is somehow less. But in the in vitro assays, actually, you see that both are affected. And I was thinking maybe in the direction of more like half-life of bone or something like that.
Yeah, indeed, it could be the length of time that these mice have been treated with glucocorticoids, 3 weeks. It might not be able to see those effects. Yeah. And obviously, we have direct versus indirect effects in vitro and compared to the mice. So, well, it's easy to sort of add a component onto osteoclast or osteoblast and see what the effect is, what's happening, obviously, in vivo in the mice. It could be a completely different story with the indirect effects of the inflammation.
Right, thank you. Yeah. Is there still time for a question or a very short one? Yeah, well, then I'll also make a very short question for probably a very short answer. So did you, but then probably answers no, also look at the cartilage actually of these mice, because that is actually my hobby, but there's no cartilage analysis whatsoever, while of course also, well, not only osteoarthritis, which is my thing, but also rheumatoid arthritis, there is massive degradation of cartilage, and I was wondering whether you saw any protective effect actually of the glucocorticoids or negative.
No, I'm not too sure. I don't think that was something that we fully investigated. However, I think it would be something that would definitely be worth looking at, even from the muscle side as well. I think it would be important. So, yeah.
Thank you. Okay.
Thank you.
I give you the word back.
Thank you. The opposition will be continued by Professor Jonkers. She is a professor of intestinal health at this university and today's secretary.
Thank you very much, dear candidates. Congratulations with this very interesting and excellent thesis. I read it with great pleasure. I was also quite honored that you pointed out inflammatory bowel disease in the first of your chronic inflammatory disease. I think it was by chance, but it was nice to note. I think also it's really nice that you really go into the mechanism, because I think that's really the way to try to identify preventive or therapeutic strategies to hopefully intervene in the future with this muscle wasting, etc. I also try to learn from all your findings and the nice reviews in your thesis for inflammatory bowel disease. I know that it's not your area of expertise, but I think there's a lot of overlap with the diseases you studied. And to start with, there's a wide range of cytokines involved there, like TNF-alpha, IL-1 beta, interferon gamma, a lot of IL-6, IL-8. I can mention them almost all, but it also varies between patients. We also noted that there's in a substantial group of IBD patients, there's also a low muscle mass. I have to say, we really lag behind in IBD to learn from what you already know in COPD, etc. So I would like to understand a little bit whom of our patients are at risk. So maybe you can help me out a little bit with trying to elaborate a little bit on what is the role of, for example, TNF-alpha then in comparison with other cytokines, or are they all as important if we look to muscle wasting?
Okay, thank you, the highly esteemed opponent, for your question. So in regards to the inflammatory cytokines and their importance in muscle wasting in humans, as like you said, described in the in chapter 2, they are a number of cytokines that come up in many different patients, in COPD, IBD, chronic kidney disease. I think chronic heart failure was included in there as well. There is, from what my understanding is, there is a particular cytokine that I think is a focal point, which is TNF-alpha. It's an apex sort of cytokine that a lot of the time drives the inflammatory cytokines like IL-6, IL-1 beta to be upregulated. However, as discussed in the in the studies when we've looked at mouse models of these diseases or chronic inflammatory disease or even just inflammation in itself, we've seen inhibition of these cytokines having a good prognostic outcome for the muscle wasting, but it wasn't very translatable in humans.
Yeah, I was indeed wondering, because I'm quite happy that you mentioned the TNF-alpha, because they consider it as a key driver in IBD, and I think about maybe over 1/3 of the patients are now treated with anti-TNF-alpha.
Yeah.
So, do you think it would help against muscle wasting in these patients?
I mean, the short answer is no, not really, and that is why obviously the basis of this thesis is looking at alternative potential therapeutic targets like 11-beta-HSD1 or glucocorticoid activating enzymes, because the inflammation, you're obviously going to have the inflammation driving the muscle wasting, but the resolution of the inflammation therefore is important, and we know that glucocorticoids are very good at directing their eliciting their direct anti-inflammatory effects on the resolution of the inflammation. So I think, in a short answer, I don't think anti-cytokine therapies are going to be as effective at suppressing inflammation as what we already have, which is, NSAIDs, glucocorticoids. Okay, the clinicians really like to use the anti-TNF therapy, and so I really hope that it also would help a little bit. But if it then, because IBD is very complex, and I also learned that in chapter 3 you used LPS to induce these exacerbations, and I can understand that because it can lead to cytokines, etc. You come back to the inflammation. But if we look to IBD patients, then we also see quite a lot of LPS in the circulation because they have a disturbed intestinal barrier function. Could the LPS also have a direct effect in the muscles on muscle wasting? Could it also have a contributing factors? Could it be a contributing factor in these patients, do you think? The LPS directly?
Yeah.
I think there's both direct and indirect effects happening with the LPS. I think the intratracheal installation in this particular model, obviously you have this cytokine storm in the lungs to start with, but then that spills over systemically, and then so I think it has an indirect effect on the muscle wasting. But you can never be sure if it is a direct effect. There has been previous studies been done in our labs where they've had the LPS treated animals without emphysema. And they saw muscle wasting as well, but again, is that due to the LPS an indirect or a direct?
Yeah, it came to my mind because I did read your chapters carefully because I learned a lot from them. And in chapter 2, you also mentioned the TLR4 receptor, for example, and that could have direct effects. And then there are also these receptors in the muscle. So I was thinking, maybe that could be something, could be interesting for us to explore there. And then I have some time left. And then the patients also often suffer from stress. You can imagine if you have to go to the toilet all the time, it is quite stressful in your life. And then we also see that stress really results in exacerbations. Should we also consider stress an important factor in muscle wasting for these patients?
Oh, absolutely. Obviously, glucocorticoids are increased with stress, but then inflammation, low-grade inflammation, the resolution of those inflammatory processes may be diminished slightly if you've got an elevated HPA axis, constantly stressed, constantly having it might dysregulate the HPA axis, which then may mean a dysregulation in the release of endogenous glucocorticoids, not helping to resolve the inflammation. So again, I think there is definitely indirect effects at play at work, but absolutely, I think stress would definitely, in all of these patients with
I think it's really interesting for us to look a little bit deeper into these patients and to see how we can understand who is more at risk for muscle wasting than others. And maybe a final question, if I am allowed to, because you mentioned the glucocorticoids already yourself. And also in our patients, even up to 50% of the patients receive glucocorticoids. And I think you nicely showed the relevance of that for muscle wasting, but also of the enzyme and inhibiting that. But I also was a bit confused on the results of chapter 3 versus chapter 6 and inhibiting this enzyme. And so maybe you can help out there a little bit further on the seemingly contradicting findings you have on inhibiting this enzyme with regard to muscle wasting.
Yeah, of course, thank you. It's a very good point. So the crux of chapter 3 is looking at the endogenous level of glucocorticoids, and obviously chapter 6 is looking at the therapeutic levels. So chapter 3, when we're looking at how the endogenous levels are modulated with the 11-beta-HSD1 gone, we do see this increased muscle wasting. But then when you combined the glucocorticoid treatment with the 11-beta-HSD1 knockout animals in rheumatoid arthritis, we saw this partial protection. So it all stems down to glucocorticoid excess. There's been previous work done where they had 11-beta-HSD1 knockout animals that were treated with glucocorticoids outside of any inflammation, and they were protected with muscle wasting. And with the acute exacerbation model, there's also looked at that in GR inhibitors and knockouts, and saw that was protected. So that was the hypothesis of why we thought it was going to be protected. But in hindsight, there has actually been other studies where they looked at endogenous levels of glucocorticoids in the 11-beta knockout mice with rheumatoid arthritis, and that muscle wasting was worsened. So comparable, they do match up. But so in theory, if you then repeated chapter 3 with therapeutic glucocorticoids as well, we would hope to see what we see in chapter 6, where you had a partial protection.
Okay, and do you see maybe final last short question? Do you see potential to target this enzymes in the patients to really try to prevent the effects of glucocorticoids? Could there be a therapeutic strategy for the future?
Yeah, I think of course there are actually 11-beta-HSD1 inhibitors in stage 2 clinical trials already, which have been used for diseases such as diabetes, and so far they've been quite promising, but they've not been really utilised in the way that it has here with muscle wasting. So I think a key point of the reason why it would be good with muscle wasting is because 11-beta-HSD1 is found in such abundance in muscle, and it's not found in abundance in many other tissues. I mean, it's found in abundance in bone and adipose tissue as well. So with that in mind, it's sort of like direct target therapy without directly targeting, where you could still possibly use glucocorticoids to systemically alleviate the inflammation, but then have this like 11-beta-HSD1 inhibitor stopping the activation within that muscle.
Thank you. I'm really very pleased with the answer, and I see that the pro-rector wants the word back. So thank you very much.
Thank you.
Thank you. The opposition will be continued by Professor Breen. He is a professor of metabolic and molecular physiology at the University of Birmingham.
Thank you. Hi, Justine. Can you hear me okay? I hope so. Yeah. Congratulations again on I can see myself twice now. This is very strange. Congratulations again on a really nice thesis and a great presentation as well. It doesn't seem that long ago since we were last speaking about the work. And obviously the last time we spoke was maybe 2 or 3 hours. So I think I exhausted most of my questions then, but I thought of a few additional things to ask you today. So the first thing I want to touch on is something related to your general introduction. There's an emerging debate around the contribution of local versus systemic inflammation to muscle wasting in conditions of inflammatory disease, cachexia. And of course, that may differ with different underlying etiology, but it also may differ between animals and humans as well. So either from your background reading or on the back of the experiments you've done, can you comment a little bit more on what you think is really driving this rapid muscle loss in patient populations and how that differs to inducible models of inflammatory disease?
Okay, thank you to the esteemed opponent for your question. So just to confirm that I've got the sort of question right, is elaborating a little bit more on the differences we may see in human populations and animal models in direct and indirect causes.
Is local inflammation really a thing in humans?
Sorry, I didn't catch what you said there.
Oh, sorry. Is a local inflammatory factors really playing a role in this rapid muscle wasting in humans, or is it largely driven by systemic inflammation?
Thank you. So yeah, I think both. So in order to have that systemic inflammation, as I said previously, that you have this inflammatory storm localized in, like, for example, RA patients in the joints and in COPD patients in the lungs, and then this inflammatory storm is what causes it to overspill. So it would be fantastic if we could just alleviate the risk of it overspilling into systemic. I think that would reduce a lot of the extra articular, extra pulmonary consequences of these diseases. But unfortunately, I'm not really sure how you would do that in theory in humans. So I think you can't have one without the other, but they're both very significant. Yeah, I don't know if that answers your question, but yeah.
Yeah, in humans, is there any evidence that inflammatory factors, signaling mediators, are upregulated or dysregulated?
Yeah. Yeah, of course. So when you look at human cohorts with lots of different diseases like cancer cachexia, you see these upregulations of like CRP, IL-6 in the serum. You also see the infiltrates of the inflammation locally as well. So I think it's definitely if not a direct driver, an indirect driver affecting the other, like I said before, like HPA axis, affecting glucocorticoids, affecting what was outlined in, I think it was chapter 4, the indirect effects of inflammation on all sorts of different systems within the body. So yeah, I think it would be both that would need to be tackled, but I think you do see in patient populations this systemic increase in inflammation being correlated with muscle wasting.
Moving to chapter 3, I can't remember whether we spoke about this or not a year ago, but some aspects of this paper seem to have been rewritten. I guess for publication, it seems like it's under review at the moment. Obviously, you've got this kind of surprising find, somewhat surprising, I assume, that muscle wasting was exacerbated in the LPS 11 beta HSD knockout animals compared to the wild types. You do go on to offer some hint of an explanation as to why that may be and why that discrepancy may be there in comparison to previous studies. I didn't feel like you really went into that in lots of detail though, so I'm wondering whether you can articulate that a little bit more for me now. Why this surprising finding, and then what do you really think is driving that?
Yeah, thank you for your question. Just to highlight, this particular paper has actually been accepted for publication not too long ago. So it's even different to what you read now. So that is fantastic news on our end, yeah. In regards to the potential reasons why we see this increased muscle wasting in these knockout animals, one potential reason is the dysregulation of the HPA axis. So obviously 11 beta HSD1 is found in the brain, is found in areas along that HPA axis, and without that negative feedback loop, say in too many glucocorticoids, you don't have that negative feedback loop, and therefore you may not have, therefore you may have more glucocorticoids being released as a result of that. So that was one of the ways we postulated, and obviously this correlates with the fact that we did see elevated levels of glucocorticoids, of corticosterone in the serum of these mice that were had acute exacerbations but had no enzyme. So that was one of the primary ways that we think that it's driving these changes in the muscle.
Right, thank you. That's nice and clear. Thank you, Justine. Again, I think I've got a very short question, and then maybe a longer one to finish with if I've got the time. It's stated throughout this paper, and I don't know if this has changed in the final version, that muscle wasting was exacerbated in the the LPS knockout animals. But when I look at the data, as I did maybe a few months ago, it doesn't seem to indicate that there is a kind of interaction difference there in the extent of loss between the groups. Can you speak a little more to that? And I guess what was the final story there, or am I misinterpreting something in that? I'm looking here at the delta change in muscle mass in figure 2C in that chapter. So obviously there's quite a significant magnitude of difference there, but I'm not sure from the indicators whether it differs between the groups.
Give you a question. So, yeah, indeed, we did discuss this during the Birmingham Viva. Yeah. So I think one of the primary reasons that we see such delicate subdued effects is, unfortunately, this study was slightly underpowered by the end of it due to the dropout that we had in the animals. However, the effects that we see in regards to when we looked at muscle gene expression, we saw that there was discrepancies between the amount of induction in the atrophy gene expression compared to wild type and knockouts. Anti-anabolic expression was lessened in the wild type. But in regards to looking at different muscle groups as well, so for example, we do mainly focus on gastrocnemius, but we also saw in soleus and a couple of others, the muscles that we took, that there was exacerbated muscle wasting in these particular muscles in the knockouts compared to the wild type. So I think it really depends on, it would be really interesting, and I would like to really look into the fiber type changes in these muscles. So is the atrophy affecting particular fiber types? Are we seeing changes in the fiber types? Yeah, I hope that answers your question.
I think it will be really good to perhaps in future experiments, and maybe this is something Rowan and I can speak about, is to try and mimic a challenge, an anabolic challenge on these muscles as well, to see how they respond and if that anabolic resistance is present, and then perhaps that's what's driving some of the changes that you see here in muscle morphology. Final question from me is, I guess I'm just interested, really curious to get your take on, beyond pharmacology, what the frontline lifestyle therapies are for chronic inflammatory disease patients at the moment, and is there a way that lifestyle therapies, alterations in exercise or diet might interact with glucocorticoid therapy, for example, or molecules that can effectively dampen down 11-beta-HSD. Tell me your thoughts on that.
Thank you. Very interesting point. So in regards to lifestyle effects, I really do think it depends on the disease type we're looking at, how the disease progression is in that particular person. Somebody who's in stage high gold stage of COPD is going to be very limited in what they can do lifestyle wise compared to somebody who's on a lower stage. In regards to sorry, I'm just trying to formulate my answer again because I had it in my head. But in regards to lifestyle changes, so we know that, for example, cachexia in a lot of these diseases is driven, and that increased exercise and increased protein intake doesn't always mediate the loss of muscle in cachexia. So again, it depends on the population or the person specifically. There might be maybe one day have the ability to adapt specific changes in lifestyles in combination with therapies to different people who react differently to those. I don't know if that's formulated in the way that I wanted to, but do you see? I don't know if it's.
A good focus. I'm thinking about if we go back to pharmacology as well. I'm thinking about the potential for hormone replacement therapies or myostatin knockouts and myostatin inhibitors to have a potentially supportive effect here. But I guess you always run the risk of dosing patients up too much with too many therapies and too many side effects is always something you want to avoid. So yeah, great. Thanks, Justine.
Thank you. The opposition will be continued by Professor Franse. He is a professor of personalized management of COPD at this university.
Thank you, Pro-Rector. Dear candidate, first of all, let me congratulate you with your thesis and also the congratulations to your team. I really enjoyed reading it, and I must say that I often make the comment that basic scientists should be more involved in the clinic to understand what inflammation and COPD exacerbation is, but I came to realize that actually I should do the opposite as well, because I learned a lot of basic research from your thesis. And also I'd like to congratulate you on your cover, which is, I think, very optimistic, because I see perfectly healthy lungs and I see a not very cachectic woman there, so that is very hopeful for the future. I'd like to discuss with you your comment regarding chronic inflammatory disease. Because in your introduction you say, well, chronic inflammatory disease is an umbrella term, and you use several examples. Also in your presentation you use rheumatoid arthritis, COPD, heart failure, kidney disease. And it made me actually wonder whether I should be not a pulmonologist, but an inflammatologist or something like that. Is it really true that these diseases are so common that you can actually call them in one line? That is my first question. And the second is, I'm missing a very important inflammatory pulmonary disease, asthma. Because in asthma, inflammation is part of the definition of the disease, and in COPD it's not. So why is asthma different, and why was it not included here? And
thank you to the highly esteemed opponent for your question. So the first question in regards to it was the umbrella of chronic inflammatory disease and inflammation. So indeed, a lot of these diseases hold several factors that are very similar within each other. But so an umbrella term, I think, is a very loose term for it, and I don't think focusing on all of them as being able, because they have such different pathophysiologies and they do have different like consequences systemically. Although some of them overlap, there are different factors at play. There are different, for example, COPD, you've got reduced lung capacity, airflow limitation, and this is obviously could lead to acidosis and hypoxia, whereas you may not see that in inflammatory bowel disease. You might not see hypoxia as a side effect because it's not affecting the lung and the airflow limitation. So I think it's when we're looking at a particular disease in regards to potential therapies and how these pathophysiologies of those particular diseases are, you know, how they continue. To put them all under the same umbrella as one will work for all for one, I don't think that is fair to say.
Okay.
So I do think it's important to make sure that we're looking at specific diseases underneath that umbrella, but remembering that they do have things that...
Different pathophysiologies.
Yeah.
Okay. And what about asthma?
So in regards to asthma, we do know that obviously it's a lung disorder that is affected with inflammation. As far as I'm aware, and I'm not sure 100% on this, so don't quote me, but it doesn't have the same destruction of the airways as you see in COPD. Mm-hmm. So we don't see the alveolar breakdown to quite the extent that we do, or the chronic bronchitis as we do in COPD, as we do in asthma.
Yeah, but we see a lot of airway inflammation in asthma.
Yeah,
I agree with you that the destruction is not a characteristic of asthma, but but why could it be that this kind of inflammation in the airways does not result in muscle wasting? Or
no, indeed. I mean, it's a very fair point, and there is no reason to believe it wouldn't. So I think I would like to see a similar sort of study with the enzymes and the glucocorticoid therapies, because obviously glucocorticoids are widely used for asthma patients, and see how that corresponds with the data that we have for the exacerbation.
Could it be that asthma is a different type of inflammatory disease, different interleukins, different cytokines involved, but also eosinophils as a main driver and not macrophages or neutrophils.
Yeah, of course. Yeah, the infiltration of the different types of cytokines in the lungs.
Yeah. And that relates to my second question, if I may, because I was intrigued by your model. You use LSDs treated mice, they develop emphysema, and then you trigger them with LPS to have an exacerbation. Mm-hmm. But how representative do you think that is for the COPD population we see in the clinic?
I think it's a very interesting point. So I think in regards to how representative it is probably quite a small, not a small window, but a smaller window than the overarching story. You have several risk factors that are associated, like smoking, of the development of chronic bronchitis and emphysema. And then you also have viral infections that can induce the exacerbations and other triggers. So I think it would be fantastic to look at all of those different developments and the different ways in which COPD and acute exacerbations develop in context of the 11 beta. Mm-hmm. However, this particular model was of interest to us because it had the muscle atrophy associated with it, mm-hmm. which is really what we wanted to have. We wanted the systemic effects of induced muscle atrophy, which you may not see in other models. I'm not too sure, you probably do, but I think for this particular research it would be
so you actually say, well, this is an emphysema model, mm-hmm. not necessarily a smoking COPD model, and you say it's maybe mainly reflects the bacterial exacerbations and not necessarily the viral ones. Mm-hmm. Yeah, I agree with you. We also see different, apart from the bacterial and viral exacerbations, we also see exacerbations caused by eosinophils. And we also see exacerbations of patients without any evidence of inflammation. Mm-hmm. So should we start a new research, for example, to look whether different type of exacerbations are at different risks for developing muscle wasting?
Yeah, of course. I think it would be really important to highlight which patients are more at risk. So we know, for example, emphysematous patients are more at risk to muscle wasting than those that have chronic bronchitis as their primary feature. But again, in regards to exacerbations, as previously done in our lab, the number of exacerbations is also important in muscle wasting. So previously done as frequent exacerbations and the lack of muscle recovery after these exacerbations is important to look at. I think there's a lot of many different pathophysiologies in these diseases, and in order to really pinpoint what's happening with the muscle, we would really need to look at everything as a whole, but it's very difficult to do that.
I have one final question, if I may, and I'd like to refer to proposition 8. Maybe one of your paranymphs can read it out.
Yeah.
Sure. Hello. Effective treatment of muscle wasting will reduce exacerbation frequency of chronic obstructive pulmonary disease and disease flare-ups in rudimental arthritis.
Yeah. So I was really intrigued by that one. Do you think it's correct? Or did you make a mistake?
No, I think it is correct. I stand by it. I think it is a predictor of exacerbations and flare-ups, muscle wasting that we see. However, you don't always see muscle wasting in these patients.
But I mean, we just discussed the causes of exacerbations, mm-hmm. Bacteria, viruses, eosinophils. How can you change that by reversing muscle wasting? I don't see that.
I don't think you would be able to change the I think it's an interesting point. So I think you obviously have the correlation between increased muscle loss with the increased risk of exacerbations. And I think muscle wasting is often not acknowledged as important as it is. It is, like I said in my presentation, it's a hugely important role in homeostasis of the body. It's 40% of total body mass. I think that generally it's an undervalued
With that, I totally agree.
Thank you.
Thank you very much.
Thank you. The opposition will be continued by Dr. Gosker. He is an associate professor of molecular muscle metabolism at this university. Dr. Gosker.
Thank you. The candidate, congratulations with the nice work and the nice thesis. I also want to congratulate, of course, the team. I want to discuss a little bit on chapter 3 with you. In summary, you have this exacerbations of CPD model where you induce emphysema with elastase, then you induce an acute exacerbation with LPS, then these mice develop muscle wasting. And if I am correct, the idea is that this muscle wasting then, that was your hypothesis, would be at least partly mediated by the endogenous corticosteroids. And by knocking out this 11-beta HSD1, you thought to prevent this. But that didn't work out as you thought. And you found the opposite, so the wasting was actually aggravated, and you also saw an increase in circulating corticosteroids, right? So my first question is about the model. Why did you go for a global knockout?
Okay.
And would you have still done that?
Yeah. Thank you to the esteemed opponent for your question. I love getting this question. I, of course, would always go for a global knockout. And the main reason for this is because the translatability from mouse to human is a lot easier done. I mean, if we wanted to look at what was really going on in the muscle, a muscle-specific knockout is absolutely imperative to really look at the finer details of the different pathways that are affected, etc. However, when we look at the translatability aspects of that, what kind of inhibitors, 11-beta HSD1 inhibitors specifically for the muscle would be able to be found on market? Whereas if we have these mice that have a global knockout of the enzyme and 11-beta HSD1 inhibitors that are being developed and in clinical trials, this translatability, I think, is more so.
Okay, so if I understand correctly, the reason why you chose for that is because when you translate this to clinics, you will give some drug or medication, and then it would be impossible to locally target this enzyme. Is that what you're trying to say?
Yeah, that's what I believe.
Yeah, I agree. But from the scientific point of view, it would be, of course, interesting to go for a local knockout, because of course you have this trade-off. You also described this in your thesis between trying to reduce corticosteroids in the muscle versus combating the inflammation. So in that sense, you don't want to reduce corticosteroids globally, I think.
Yeah, of course, and I think that's why it's important that we do the in vitro and ex vivo experiments as well, because whilst we have the in vivo looking at the direct and the indirect effects of the inflammation and 11-beta HSD1 inhibition wholly, when we have these like ex vivo muscle cultures, in vitro muscle cultures, where we look at the direct effects of inhibition and inflammation and glucocorticoids, that's where you can really look at what's happening. And obviously we do want to reduce the amount of animals used in research, so I think that's why in vitro and ex vivo cultures are very important.
Yeah, okay, I agree. Then a little bit more into the muscle wasting. I think what you're trying to say with your figures and the data is that because there's no real difference between, so the glucocorticoids are increased, but there's no difference in inflammation measured by IL-6, and also no differences between the wild type and the knockout in inflammatory signaling in the muscle.
Yeah.
And based on those data, I think you kind of suggest or conclude that inflammation doesn't explain why wasting is aggravated in a knockout.
Yeah.
Are you 100% sure? Because you only measured one cytokine, and you just mentioned TNF-alpha, for example, as the apex predator. And also in the muscle, I think the markers that you use to measure inflammatory signaling mostly represent the NF-κB pathway, and there might be other pathways involved, inflammatory pathways involved, as you show in figure, a nice figure in your introduction chapter. So can you comment on that?
Yeah,
Could there
of
be
course.
other inflammatory processes still that you didn't look at that account for the wasting?
Yeah, of course. So we did obviously look into more of the inflammatory gene expression than is outlined in the paper. So there is a lot of data that is unpublished data of inflammatory cytokines, CXCL1, CXCL2, IL-1, IL-6, a plethora of different cytokines that we looked at.
But did you measure those in this study?
Yeah, it was a unpublished data that we've got as well, and they all showed similar, similar inductions of inflammation in both.
So no difference between the wild type and the knockout?
Yeah.
Okay.
Across the board, obviously with an exacerbation, which is one of the primary reasons why we didn't believe that inflammation was a primary driver in this instance.
And that's circulating cytokines, but then in intercellular signaling, so you had some markers of NF-κB signaling, I think, but what about, for example, JAK-STAT pathway could be involved in wasting as well? Did you look at it as?
Yeah, I think so. I think it would be interesting to have a look at the NF-κB signaling in a bit more of a focal point to determine if, because obviously we know that in the previous models within the group that NF-κB has been potently upregulated in response to acute exacerbation of COPD. So I think it would definitely be something worthwhile really looking into.
So, but if I'm correct, from let's say that you do that and that similar results come out of that, that then a conclusion could be that inflammation or differences in inflammation could not explain the aggravated wasting. So then you would attribute the aggravated wasting directly to the glucocorticoid signaling, I guess?
Not necessarily. I think it depends on the timing as well. So obviously we've noted that this particular time point that we've looked at is 48 hours post LPS, and does that mean to continue? Oh yeah. So so we look at the 48-hour time point, and then we know at that point is when you start to see muscle recovery. So this could be that the point in time that we were looking at, inflammation was already resolved to some degree, whereas it would be really interesting to look at 24-hour time point and see if inflammation was a key driver in the wasting, and then the glucocorticoids were alleviating the recovery. So I think that would be a good step.
Okay. Thank you.
Thank you. So, Justine Webster, the time appointed for defending your thesis has now passed. The degree committee will withdraw to discuss the quality of your thesis and of your defense. I request that you and your company await our return and the outcome of our deliberations in this hall.
All right, please.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes.
Long road to the south side. 7 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. All your time, 'cause I'll go the extra mile. Long road to the south side, 7 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. On the dash, got my laces tied. Long road, I don't waste no time. Break rules because fate decides. With the team and we chase the light. I make a move, fall down, shake it off. I hate to lose, bad branch, break it off. No room for negativity, praise and love. Prepare for deep part cause we're taking off. Hit the mileage, traveling so far from home. Gathering knowledge, other connections. I put the time in to get everything I ever hoped for. It's a challenge and we all in. It's a better future that we go for. And even though these nights seem long, I know that I am never on my own. Know you've won the battle once you've grown. Outside, 10 miles in my rearview mirror. I know what it felt like. My goals only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and die. Long road to the south side. 10 miles in my rearview mirror. I know what it felt like. My goals only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and die. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile.
all smartly dressed. Smartly dressed.
How come he didn't speak?
Because he's my supervisor.
Oh, he's
and
just
he's like Ramon.
he's just looking in.
No, well, I think at the end they do like a little speech each.
Okay.
But they don't get to ask questions because it's like they've guided me through it. So them asking
Of
a
course,
question,
I understand.
they
Yeah.
might ask me a question that I've told them.
Would help you.
Yeah. So, aye.
That's an impressive gobble.
This was such a good buy. Did you notice I was messing
I
with
didn't
it?
notice. No.
Yeah, I was messing with it the whole time. I was literally like...
I was trying to look and I was like trying to nod with you and stuff like that.
Yeah, crayons.
What a great point.
Oh god, I'll be like, okay.
I know.
There we go.
Should we stand?
Yeah.
I think they say all stand.
This is just the fun part, so you don't have to be nervous. I'm nice.
Justine Webster, the degree committee here present has assessed the quality of your thesis and your defense. In view of its positive verdict and taken into account your previous qualifications, the degree committee has decided to grant you the degree of doctor. Professor Scholz is authorized to confer upon you this academic distinction in accordance with Dutch university custom. I invite your supervisor to now take the floor.
All right. Do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent, and responsible?
Yes, I do.
By the authority vested in us by law and in conformity with the decision of the committee here present, I hereby confer upon you, Justine Michelle Webster, the degree of doctor and grant you all rights attached by custom and law. As evidence of this, I now present you with the degree certificate signed by the rector, the secretary, and the other members of the committee, and affixed with the official seal of the university. Justine, really congratulations for this distinct award. It's really incredible to have your PhD defended today in Maastricht and previously in Birmingham. Not many people have this distinct. Yeah.
Thank you. Thank you very much.
Whatever it is. But, and I also would like to congratulate your proud mother. But now I quickly hand over the microphone to you both co-supervisors, and starting off with Ramon.
Thank you. Dear Dr. Webster, I'm still trying to figure out how to deal with the double doctor degree that you just obtained, but dear Justine, here you are at the final stage of your PhD journey. And that journey started in 2017, when our proposal with Dr. Hardy was awarded as part of a competitive funding scheme to stimulate collaborations between Birmingham and Maastricht universities. The only thing we still needed was an adventurous candidate to join our teams. And following interview in person with Rowan and a very old-fashioned telephone interview with me, no Zoom present at that time, you were selected. You started in 2018 in Rowan's lab in Birmingham. Skype was regularly used for our bi-weekly meetings, and there were always very lively and joyful events with you and Rowan crammed behind his camera at his tiny desk. Despite all the uncertainties that Brexit brought along, you came to Maastricht in March 2019, just missing out on Carnival. But with your residence close to the center of Maastricht, you were in a good place to celebrate it the next year, or so we thought. In the lab, you really prospered. You rapidly mastered the techniques in our laboratory and also introduced new ones, including refined mouse biotechniques for which you and Natasha were trained in Utrecht. It was impressive to witness the agility and the speed by which you acquired these complex skills. You also attended international conferences that year, including the Cachexia meeting in December 2019 in Berlin, during which you received the Best Poster Award. This type of international travel dramatically stopped, however, in March 2020, when the COVID pandemic overwhelmed Europe and the first lockdown was enforced. Fortunately, you had decided to return to the UK just a few days earlier, which in hindsight was a very good decision. This meant, however, that your ongoing massive mouse study had to be completed during a couple of 16-hour working days by Sarah, Wouter and me. The second and still unresolved problem, however, is that you missed Carnival again. In the following period, part of your lab work was conducted by remote supervision of Kelsey, who, as an intern, together with our lab technician Marco, performed the analysis described in Chapter 3. With Laura, another master student, you produced Chapter 2, by now a highly cited review article. In parallel to all of this, you completed your thesis, culminating in a successful PhD viva in Birmingham. Considering your determination and your perseverance in overcoming the challenges that you faced during your PhD trajectory, I feel you should be extremely proud on the thesis that you produced. I can say for sure that Annemie, Rowan and I certainly are. Therefore, my sincere congratulations to you, your mother and your friends. I wish you all the best at the personal and professional level and hope you will enjoy the rest of your time here in Maastricht. And with that, I would like to give the word to Rowan as the other co-promoter who was also instrumental to the productive and fun time we had together. Rowan, the word is yours.
Thank you very much, Ramon. Wow, what a fantastic event it's been today. I'm so happy to have seen the whole process, and it's been truly amazing. Justine, I'd like to say congratulations on your amazing achievements during your time with us over the last it's coming up to 4 or 5 years now. I think Ramon highlighted those points of when we were looking for your position to fill, and it was a tough time actually. We were looking outside of the normal recruitment period, and it was a challenge finding a student. But actually, we traveled around, and I remember going to Liverpool to highlight the need for a really high standard student, and it was your name that came to the fore from other people that I spoke with. And when you interviewed for the post, you really stood out head and shoulders above the other candidates. The things that sort of made you stand out, your motivation, your enthusiasm, and your clear excitement and passion for the research really came out. And it was really good, a really great opportunity for us. And I hope for yourself when you chose to come and take up the PhD. More so that you were brave enough to travel down to Birmingham to do it. Not always the easiest place to attract students to in the UK, but you did come, and you came as a raw talent. You worked incredibly hard. You were quick to learn. And I think one of the things I'd always say about you is that whenever we set you a task, whether it was something you knew what you were doing in advance or whether it was something entirely outside of your comfort zone, you got the job done. And you always worked hard at everything we gave you to achieve. That included all the other aspects of your PhD, whether it be the social ones and the partying, or whether it was the academic networking. And I think very fondly back to our times of many conferences and lunch breaks and a few of the conferences, late night parties and 2, 3 in the morning, still enjoying ourselves. And I think the key thing there was you were always awake the next morning to deliver your presentations and talks without any complaint, which is always another great sign of any academic. I think I've seen you grow a lot during the time you've been with me during the PhD, and something I've always been impressed at is how you've grown both intellectually in your writing, your academic and presentation skills, but also as a person. You've developed so many fantastic interests during your time working with us. And I think back to your interest in feminist literature and politics and philosophy, and what really have made you an incredibly well-rounded individual. It's fueled a lot of fun conversation and discussion at lunchtimes and into the evenings with over a few drinks. And I think they've just been part of the tapestry that have been so fantastic. So in whole, I'd like to forget all of the complications you had during the COVID pandemic. It caused a lot of complications. It made life difficult for you, but at the end of the day, you did what you always do, and you just overcame those problems and worked through them. And here you are now, completing your thesis, 5 really impressive publications, a fantastic network community and collaborative interaction that you've helped support and build. And I think what I would say is now you're going to be very much missed from both of our labs. You've been a fantastic individual to work with. For me, myself, I almost feel of you more as family now after the amount of time you spent working with us, as much as a PhD student. And I'm really excited to go and see what you do next and where you take your career forward. So I'd just like to take this opportunity to wish you the best, say thank you so much for all of your hard work over the period of time you've been with us, and congratulations on such a fantastic achievement today.
Dr. Webster, on behalf of the entire scientific community at Maastricht University, I congratulate you with the degree that you have acquired. And should your future include research, I hope that you will be able to continue working with us in Maastricht. I wish you a nice day. I thank all of you for being here today, and with that, I close this ceremony.